Yesterday, we looked at how we can classify organisms based on characteristics of non-instinct organisms. We talked about contemporary ideas of classifying alive versus not alive. We looked at First Nations perspective, and then finally we looked at what scientists use as a criteria for something being alive or not alive. So this idea of classification is built on group of things, on a specific set of characteristics. You are going to receive items. This group of items, I have packing tape, masking tape, pencil crayon, pencil, highlighter, and scissors. I have two groups of four, since we have 8 people. As a group, I want you to discuss how you learn these items. You decide what group of 4 you are in. There's your items.
Well, let's do this right here.
Here are your items.
Ignore the middle part on your right. Should have bite it off.
Okay. How would you decide how to group these items?
We decided to group them based on what you can use them for. So we went with writing utensil, taping or sticking utensil, and cutting or carving.
Okay, so you have 3 different groups.
Yeah.
Okay. Those 3 groups here.
We have like 12.
Okay.
You said these should be categorized as they're sticky.
Black and black.
These, I said, so they're sharp. So I said, okay, here. Both of these can be sharp. Both of these could be sharp, but they can also like color. This can color here, so I kind of put them. These kind of in the middle here. It's kind of its own thing. So kind of like the.
Okay, so this group is categorized based on what you can use for. You have a bit of a mix. You have some features and you also have potential use. What could be some issues with how you group your items?
You can write with these.
Oh.
If you taped it into letters, you can write with tape. So you're very soft.
What you think an item would be used for is not necessarily how another item would be used. Okay. What else could be some potential issues?
When you think of carrots, there's also cylinders.
Okay. What if discovered a new item? Would your new item be able to group based on your classification?
Yeah. Well, it depends what the item is.
If it was something like this, what if it was something like X?
No. It would be a new group.
This is a new item. Where would it fit into your classification?
Sharp.
That happens in biology.
Sure.
We have groups of individuals, but sometimes we either find a new organism or we find out something different about that organism and we have to reclassify something. That is one of the reasons why we now have 6 kingdoms of life instead of 5 kingdoms of life. Lo and behold, we discovered about a decade ago that bacteria actually have some structural differences and genetic differences. And we took that mammalian kingdom and separated it into 2 bacteria kingdoms. And we now have archaebacteria and eubacteria. And that has been because we developed new knowledge about those things. So when we group things, we tend to base them on what we see something doing or perhaps some kind of characteristic of that particular organism. What we're going to do is look at how we classify living organisms. We're going to take some of the same skills that you're using here and look at how do we treat all of these living beings that fit get those 5 or 6 characteristics of what we say makes something alive and actually group them. I'm going to show you a video, and I want you to answer 3 questions about that video. If you want, you can copy down these 3 questions, because as soon as I change the slide, you are not going to see those questions anymore. In this video, I want you to look for 3 key things. The original 2 categories of classification of living things. What was the challenge that fungi posed to the original classification scheme? And why we've had to go from 2 kingdoms to 6 kingdoms.
Described and captioned media program. In the classroom and online. dcmp.org.
Benchmark Media presents the 6 Kingdom Classification, Part 1, Animals, Plants, and Fungi.
As part of the attempt to understand our world, human beings have always tried to order and classify this vast diversity of life.
From 2 kingdoms to 6 kingdoms.
For many years, it was believed that every living organism could be placed in either one of 2 broad categories, the animal kingdom and the plant kingdom. It was a Swedish botanist, Carl von Linné, in the 18th century who was the pioneer of biological classification. Von Linné, known as Linnaeus, proposed methods of classification based on form and function that we still use widely today, although our understanding of the natural world is vastly greater now than it was in the mid-1700s. Back to him a bit later. In the 19th century, the 2-kingdom system was defined more scientifically. Each organism was carefully fitted into place, bearing in mind Darwin's new theory of evolution, which tried to show how complex animals and plants evolved from simpler organisms. The animal kingdom contained all the organisms which take in and digest food and usually move around to obtain it.
A circular diagram.
While the plant kingdom contained all those which remain stationary and manufacture their own food by photosynthesis.
A mushroom.
But fungi presented more of a problem. They remain stationary like plants, but absorb organic material directly into their bodies like animals. Because they seemed on balance to have more in common with plants than animals, they were placed in the plant kingdom.
The kingdoms are defined by circles.
When optical microscopes appeared on the scene, biologists discovered a wide variety of single-celled organisms. Some, like desmids, remain stationary and photosynthesize like plants. These were classified as single-celled algae or simple plants and were placed in the plant kingdom.
Their circle adheres to the plant circle.
Others, such as amoeba, take in food like animals, and so they were classified as protozoa, from the Greek word first animals, and were placed in the animal kingdom.
The protozoa circle extends from the animal circle.
But some single-celled organisms have the characteristics of both animals and plants. Euglena usually manufactures its own food by photosynthesis. When it's deprived of light, however, it then takes in food like an animal.
A tube-shaped euglena.
So some biologists classified Euglena as a protozoan and placed it in the animal kingdom. But others said it should be classified as an alga and placed in the plant kingdom. It was impossible to decide who was right. Then some organisms were discovered that were many times smaller than...
The Described and Captioned Media program provides services designed to benefit students who are blind, visually impaired, deaf, hard of hearing, and deaf-blind. These services include a library of free-loan described and captioned education.
So, if you take a look at the questions here, the original categories included
Plants and animals.
plants and animals. Think about a couple of days ago when I asked you to do a free write about living things, and we actually kept track of the categories of living things that were represented in our pyramids. Generally, when we tend to think about living things, we identify plants and animals because they're what we can see, they're what we eat, they're what we generally interact with on a day-to-day level that we notice. So the original classification schemes included plants and animals. Lo and behold, we started to discover things such as euglena, such as fungi. What was the challenge of trying to fit those into an existing scheme?
They matched both the plant and the animal scheme.
When we get more things, some of them didn't fit either one. And so we had to expand our grouping in order to be able to include those. Why? If you take a look here, this was the original classification, plants and animals. And as we stepped through time, what we saw was more and more division into different kingdoms because of the things that we discovered about organisms. So in the 1800s, Protista was introduced to deal with that euglena, the organism that didn't seem to fit the plants or the animals, went into protest. To this day, Kingdom Protista is a really unusual kingdom. It's actually easier to talk about what are the distinguishing features of the other five, because Protista seems to be the kingdom that organisms get put into if they don't fit the other five. You end up with this kingdom that has a very unusual members. You have members that are like plants, you have members that are like animals, and you have members that are like fungi, but they don't actually fit into those three kingdoms. Really, really unusual. Then in the 1950s, we start to get the technology to be able to identify and categorize bacteria. So we see an introduction of the Monera kingdom, which is the bacteria kingdom. And then we get into the 1990s, and we start having the technology to see and to determine that the bacteria have unique characteristics genetically and structurally that lead into a further division. So right now, we have 6 kingdoms. By the time I teach your children in grade 12 biology, we might be at an 8 or 9 kingdom structure. This is one of the things that makes biology a very fascinating subject to study, but also a very frustrating subject to study. So, have you heard about levels of scientific classification?
Yes.
When did you study about this?
I think like maybe grade 5, grade 6,
Possibly grade 6.
or grade 5 or 6, somewhere in there.
So, in science, we have all of our living things, and those living things are divided based on a set of characteristics into 6 kingdoms. And then those kingdoms are divided into phyla based on a set of characteristics. And we keep sorting things into groups until eventually you get the genus and the species. What you're going to have an opportunity to do is to classify structures and make a hierarchy.
Stuff we need.
You will need a pair of scissors. On the side counter, I have about 3 or 4 pairs of scissors. You can commandeer the ones that you use for the opening activity. If you need to go get it from